So the first thing we're going to talk about in terms of cellular respiration is understanding the currency of a cell. Right? We talked about adenosine. Adenosine is one of our nucleotides that we talked about on Monday, and it makes up ATP, a very important medium in the body for exchanging energy. I like to talk about it as the currency of the body or the currency of the cell. This is how we exchange energy. This is how we get things done. This is how we get cellular work done. Very similar to how our US dollars can get things done in our society, right? ATP is the currency of a cell. And when we look at the structure of ATP, we talked about the adenosine, which is the nucleotide component. There's also a ribose, which is a sugar here, and then there are 3 phosphates attached. This is a triphosphate. Now, in this final phosphate here, there's some energy being harnessed between this covalent bond, right? The second phosphate and the third phosphate. In between this bond, there's energy stored there. There's lots of energy stored there. And so one of the main goals of a cell is to harness the energy between that third bond. And so breaking this bond releases that third phosphate, releases that energy, whereas putting that phosphate onto ADP stores some energy there. And one of the best ways I like to explain this is think about your battery pack, right? If you have a backup charger, you only charge it to use it, right? You don't charge it to be there indefinitely. And that's the same thing with ATP. ATP is only made to be broken. It's only storing some energy to be used somewhere else, to do something else. Okay, and so in that way, it is very much the currency of the body. And so what we're going to talk about today, and I'm sure on Friday as well, is how cells harness this energy. How do they get that energy out of this third phosphate bond? How do they take this energy and do work? How do we break down glucose to get to ATP? Right? That's the process called cellular respiration or cellular metabolism. There are 4 stages involved. There's glycolysis, there's the Krebs cycle or the TCA cycle, there is the electron transport chain, and we're going to talk about those steps in that process. And I want you to have the big picture in the back of your mind. All of the steps in this process is just to bring about ATP, just to get energy to do some type of work in the cell. And that can be different. That can be making hormones, making proteins, transporting them across the cell membrane. And that requires work, right? That requires ATP. All right, so let's get into this a little bit, how ATP is synthesized, how it's made. So ADP is this version of ATP, but it only has 2 phosphates on it. So we don't have that third phosphate. And so if we find a phosphate, and we're going to talk about 2 mechanisms for phosphorylating ADP, if we find a free-floating organic phosphate and we insert some energy here, we make ATP and we release some water. What type of reaction would this be? If we make a bond and we release water, what type of reaction would that be? Talked about this with carbs, with those saccharides. A condensation reaction. This would be a condensation reaction. We're going to make that covalent bond between these two ending phosphates, and we're going to release water, and so that's a condensation reaction. Now, again, there are 2 mechanisms to do this, 2 mechanisms to phosphorylate ADP. The first one is substrate level phosphorylation. Substrate level phosphorylation, and this is basically taking a free-floating, or taking a phosphate from another molecule. So there's some other molecule in the cell. Let's call it molecule X, and it's bound to that phosphate. And we're basically going to take it off of the phosphate and add it to ADP. We're going to make ATP, and we're going to have the other molecule without the phosphate. Okay? So in this first instance, I compare this to someone actually stealing a phosphate. Right? So the first instance, substrate level phosphorylation, we're stealing a phosphate from another molecule. And the second mechanism, or the second way that this can occur, is if we find a free-floating inorganic phosphate. So we have ADP here, and this phosphate was not bound to any other molecule. It was free-floating. It didn't belong to anyone else. And we just find it, add it on, right? Covalently bind it, and we make ATP. okay? And so these two mechanisms are going to be very important as we go on to talk about cellular respiration and where these different types of ATP synthesis occur. All right, so the first scenario, we stole a phosphate from another molecule, and the second scenario, we found a free-floating phosphate. Let's talk about how ATP is broken down. So basically, we have that energy stored. Again, this bond is only made to be broken. We don't make ATP for any other reason than to break that phosphate and release energy. And so we have ATP and water has to be inserted here. And then we break that third phosphate bond. That phosphate is released off. Some energy is released off as well. And because water was inserted here, this is a hydrolysis reaction, right? We talked about hydrolysis reactions, right? So this is how we break down or harness the energy from ATP. And as we can see, this energy that was released, this is what we're going to take to do some type of cellular work, okay? Any questions so far? Any questions? Any of this sounding fuzzy or not making sense? Please flag me down. Okay, so let's go ahead and talk about one of the most important reactions in cellular respiration, and that's glucose oxidation. Now, this reaction represents how glucose can be metabolized and energy can be released, as well as water and carbon dioxide. So if we take glucose, this is a molecule of glucose, and we add water, right? Reminding ourselves that this is a condensation reaction, or hydrolysis reaction, I'm sorry. We insert water and we end up with carbon dioxide, water, and energy. But then we also end up with a lot of energy, right? 686 kilocalories per mole. Energy is released as a part of breaking down glucose. This is why we eat. This is why we nourish ourselves. This is what we do with the glucose that our body receives. We break it down to release energy to do cellular work. Now, this reaction, again, can proceed in the forward direction or it can proceed in the reverse direction. What is this reaction? What is the process when this reaction proceeds in the reverse direction? No. Can anyone tell us? So it will be anaerobic by description, but looking for a more direct word. So when we take energy, carbon dioxide and water, and we make glucose, what does that? What in our ecosystem can do that? Photosynthesis, right? That's plants. So plants can let this reaction occur in the reverse direction by taking carbon dioxide, right? Making it anaerobic because it's not requiring oxygen, taking water and energy and yielding glucose and oxygen, okay? And in this way, plants are really important for our environment. They really support us in a symbiotic way. We create what they need and they create what we need, okay? All right, so let's continue on to look at this breakdown of glucose. So at the top here, we have our glucose and oxygen. And we're metabolizing this to give us carbon dioxide and water, and we're releasing some energy. Now we're gonna release some heat as well. This is not an efficient process, right? It's not 100% perfect. And so we don't harness all of the energy that's released from the food that we eat. A lot of it is actually lost as heat, just evaporated. All right, but most of it, or not most of it, some of it is used to make ATP, right? Again, to phosphorylate that ADP, make ATP, and that energy that's harnessed in ATP is then again broken to do some type of cellular work, okay? Once again, we're not harnessing all of the energy that's released. Most of it is being released as heat. Okay, this is not by any means an efficient process. And so some of the things that we can use this energy to do, we're running out of time. Some of those things we can use the energy to do is cellular movement, transport, molecular synthesis of hormones, antibodies, et cetera, and that is how we harness the energy to do these types of work. All right, so let's just wrap up with this final slide here, right? Just understanding that this is not an efficient process. This is going to yield us some energy to make ATP. And what's the only reason we make ATP? That's sort of the backdrop of what we're discussing here. For energy, right? We only make it to break it. We only make ATP to store that energy temporarily to break that bond again and yield that energy.